Welcome to part 14 of this video on the immune system. Okay, so we're currently in the process of discussing T-cell production in the thymus gland. And the point that we've got up to at the moment is that lymphoid progenitor cells have developed into these double positive thymocytes. And remember, there were two parts in the process to becoming a double positive thymocyte. Firstly, the lymphoid progenitor cell had to design its own T-cell receptor and produce loads of copies of its T-cell receptor once it's finished the design process. And remember, there's two parts to that. You have to design the alpha chain's variable domain and you have to design the beta chain's variable domain. Once you finish that design process, you then produce loads of copies of the alpha chain that you've designed, loads of copies of the beta chain that you've designed, and then stick them together to produce loads of T-cell receptors on your surface. And all the different lymphoid progenitor cells will produce their own design of T-cell receptor. Then the second part of the process to become a double positive thymocyte is they have to express both CD4 molecules and CD8 molecules on their surface, which is what we've drawn here. And the double positive thymocytes will at the moment still be in the subcapsular region of the thymus gland. Okay, so what's now going to happen after this process of becoming a double positive thymocyte is a selection process. Now, before I actually begin discussing this selection process, let me just motivate the selection process by talking a little bit more about what is actually going to be the purpose of this T-cell receptor and what is going to be the purpose of the CD4 and CD8 molecules. Okay, and then I'll actually describe the selection process. Okay, so let's begin with what is the purpose of the T-cell receptor. So, to talk about this, we need to go back to our discussion of the adaptive immune response. So far what we've seen of the adaptive immune response is that these antigen-presenting cells are going to present fragments of antigens from the pathogen on MHC molecules. And those fragments will be on MHC class 1 if the pathogen was an intracellular pathogen, whereas they'll be on MHC class 2 if the pathogen was an extracellular pathogen. What's now going to happen is that those antigen-presenting cells are going to go off and present the antigens to T cells, and they need to find T cells that are specifically tailored against those antigen fragments, i.e. specifically tailored against the antigen of the pathogen, that the fragment of which we are presenting to the T cell. So T cells are going to be specialized to fragments of proteins, and the way in which T cells are specialized to fragments of proteins is through their T cell receptor. So the T cell receptor is the thing which makes T cells, all the different T cells of your body, specific to different fragments of different antigens, i.e. overall specific to different antigens of different pathogens. And remember, we're thinking about the overall concept of the adaptive immune response being specific. So you're going to produce a whole bunch of T cells that are specific to the antigen. Okay, so that's what the T-cell receptor, that's a little bit of insight into what the T-cell receptor is going to actually do. It's the thing that makes T cells specific to a certain fragment of a certain antigen. Okay, now the job of the T-cell receptor is actually to bind to one of the MHC complexes, either MHC class 1 or MHC class 2, when it's got a certain peptide fragment in it. Okay, so we can understand now how we're going to actually create a huge number of different T cells that have specificities to different antigen fragments because they've all got these different T cell receptors. Right, so that's what the T cell receptor is going to do, and don't worry if it's still not completely clear in your head. It will be by the end of the video. That's just a little bit of insight into where we're going. We will discuss this in a lot more detail and with much more specifics in a moment. Now, a little bit of insight then into what CD4 and CD8 are. So, when the T-cell receptor, here in pink, binds to an MHC peptide fragment complex, it does so with another co-receptor. Okay, and that co-receptor is going to be CD4 or CD8. So let me just get up a picture of the MHC complex again. So here is a nice simple picture of an MHC complex with a peptide fragment in it. And this could be either MHC class 1 or MHC class 2. I know in this picture it's specifically MHC class 1, but it could be MHC class 2. What's going to happen is the T cell receptor is going to bind with this interface of the MHC peptide fragment complex. Therefore, its binding is going to be specific to what peptide fragment is actually in the peptide binding groove of the MHC complex. What's going to happen is the CD4 and CD8 molecules, they bind along with the T cell receptor at a different interface of the MHC. So they'll bind on this sort of an interface here, one of the side interfaces. And CD4 binds to MHC class 2 and CD8 binds to MHC class 1. So at the moment, our double positive thymocyte has the co-receptors for binding to both MHC class 1 and MHC class 2. Our T cell receptor, if it binds to MHC peptide fragment complexes at all, is only going to bind to peptide fragment MHC complexes with one of the MHC class, one of the MHC complexes. It won't be able to bind to both MHC class 1 and MHC class 2. As part of this selection process, we are going to have to decide, does this T cell receptor bind to MHC class 1 or MHC class 2? And if it binds to MHC class 1, we'll want to maintain the CD8 there, and if it binds to MHC class 2, we'll want to maintain the CD4 there, and we'll get rid of the other one. Okay, so that's a little bit of insight into where this is all going. Now let's actually get to the specifics then. So, what is going to occur is two selection processes. A process known as positive selection, so I'll just write this here. So we're going to have a positive selection process and also a negative selection process. So what's the basic principle here? So the positive selection process is called the positive selection process because we want a positive response in order for the double positive thymocyte in order to be able to pass positive selection. And in positive selection, what we're going to be testing is, can this T cell receptor on our double positive thymocyte, is it actually capable of binding to an MHC complex, either MHC class 1 or MHC class 2? Now, in the positive selection process initially, the MHC complexes that we're going to be presenting to the double positive thymocyte will have self-peptide fragments in. So we will only be looking for a very weak bond. Okay, we don't want it to bind too strongly. And you have to understand that when you create a T cell receptor that is actually capable of binding to some MHC complex with a peptide fragment, it's most likely going to be able to bind to other MHC complexes with peptide fragments in as well. It's just we hope that those bonds will be much weaker than the one with the peptide fragment that is potentially for some antigen. Okay? Right, so positive selection is all about making sure that it actually is capable of binding to one of the MHC complexes, because if it isn't, we might as well quit now. We might as well get rid of that double positive thymocyte because it's not going to serve any function at all. So we want us to check that the T cell receptor on this double positive thymocyte is actually capable of binding to an MHC complex of either the type 1 or the type 2. In negative selection, what we're going to look at rigorously is that this T cell receptor does not bind to MHC complexes where the peptide fragments are self-peptide fragments too strongly. Because if it binds too strongly, then it's likely that that T cell, if we were to let it become a full mature T cell, would then trigger an autoimmune reaction. So we want to make sure that the T cells that we are eventually putting into the bloodstream have T cell receptors that will only very weakly bind to MHC complexes where the peptide fragment is a self-peptide fragment, and therefore it's unlikely that self-peptide fragments will be able to activate that T cell. We only want to put T cells into the bloodstream and indeed into the lymphatics, and we'll talk about all of that later on, that are going to be activated by MHC complexes with non-self peptide fragments in them. Okay, so that's what negative selection is going to be all about, and the reason it's called negative selection is we want a negative response to pass. We want you not to bind to the MHC complex with the self-peptide fragment too strongly. Okay, so let's now go through this process. So we'll start off with positive selection because this is the order it occurs in. So what's going to happen now is that the double positive thymocyte that's currently in the subcapsular region here is going to come down to the cortical region, which I'm re-highlighting up here. So we'll let this be the cortex of the thymus gland. I know in my previous picture I drew it slightly higher up, but as you can see I've run out of space, so we'll have to just make do. So this is the cortex region, the region underneath the subcapsular region. So what's going to happen is our double positive thymocyte comes down to the cortex region here. And in the cortex region, there are special cells known as cortical epithelial cells. So let me draw one of these cells. So these generally have large processes as well, like so. So they generally are drawn like this. So this is my picture of a cortical epithelial cell. And the reason they're going to have these long processes is to try and increase their surface area, because that's the crucial bit that's going to be interacting with the double positive thymocyte. It's the bit that's crucial for their actual function, for their actual job in T cell development. Okay, so this is what's known as a cortical epithelial cell. So you have lots of these cortical epithelial cells in the cortex of the thymus glands. These will be in this region here. I'm just going to colour in my cortical epithelial cell. We'll have him in orange here. Okay, so this is a cortical epithelial cell, and these are the cells which are going to be taking part in the positive selection process for these double positive thymocytes. So how are they going to do this? Well, these cortical epithelial cells, what they're going to do is they're going to put on their surface MHC class 1 complexes and MHC class 2 complexes with peptide fragments from their own proteins in. So this cortical epithelial cell will produce a huge number of different proteins. It's going to chop those proteins up to make peptide fragments, and it's going to present the peptide fragments on MHC class 1 and MHC class 2 proteins on the surface. Okay, so here we go. Here are these MHC class 1 and MHC class 2 proteins on their surface. So I'll just draw these nice and simply like so. So these are representing the peptide binding grooves, and these little lines are representing the peptide fragments in the peptide binding groove. So we'll have MHC class 1 coloured in red here, like so. So that is MHC class 1 peptide fragment complex. Okay, like so, and I'll just re-straighten this up. And then this over here in yellow, it can be the MHC class 2 peptide complex. So these cortical epithelial cells are going to absolutely coat themselves in MHC class 1 and MHC class 2 complexes with peptide fragments from proteins that they have produced themselves. Now what's going to happen is our double positive thymocyte here is going to come down into the cortex, and it's now going to interact with the cortical epithelial cells. And what needs to happen is the T cell receptor on the double positive thymocyte needs to bind to either one of the MHC class 1 protein complexes or one of the MHC class 2 protein complexes. And if it does bind to one of those complexes, it will receive a positive signal, a survival signal, which will tell it to survive. If it does not bind to one of those complexes, it will not get this survival signal, and it will die by neglect. Okay, so let me actually now draw this interaction out. So let's have this as the surface of our double positive thymocyte here, like so. So here is its nucleus. Right, and let's now have this as one of these processes from our cortical epithelial cell, and now there's going to form a little complex here. if indeed the positive selection process is going correctly. So here is our T cell receptor, and now it's going to bind to one of MHC class 1 or MHC class 2. And now for the basis of this picture, let's just say it's MHC class 1. So here is my T cell receptor in pink here. And now here is my MHC class 1. So I'll just colour this in red here. And then there'll be a little peptide fragment in that peptide binding groove there. So this is what needs to happen in order for the double positive thymocyte to survive this positive selection process. After all, remember the motivation. If this T cell receptor cannot bind, at least to some extent, to an MHC class 1 or an MHC class 2 complex, there is no point in this cell existing any further. So we want to get rid of the ones where their T cell receptors are not capable of binding to an MHC class 1 or an MHC class 2 peptide complex. Okay, so the T cell receptor in this case has bound to an MHC class 1 here, and what will also happen is either CD4 or CD8 will be a co-receptor here. So in the case that the T cell receptor binds to MHC class 1, the co-receptor will be CD8. So here, this is now representing the CD8 molecule binding alongside. And unfortunately, I've previously coloured CD8 in the same colour as I've now coloured the MHC class 1. So I'll come up with a new colour for CD8. So we'll have CD8 now shown in green. So this is the interaction that's going to form. So if the T cell receptor is capable of binding to an MHC class 1 peptide complex, at least to some degree. Now, of course, we don't want it to bind too strongly, because remember, this peptide fragment is a self-peptide fragment, but we want it to bind to some degree, because if it doesn't bind to some degree, that is just showing us that the T cell receptor isn't going to recognize an MHC protein complex. Okay. And because it's bound to an MHC class 1, the CD8 will bind to that MHC class 1 next to it, i.e. it will act as the co-receptor as it's called. Whereas if it had bound to an MHC class 2, and I'll stress again that no T cell receptor will bind to both MHC class 1 and MHC class 2. They're very different. If it had bound to MHC class 2 peptide complexes instead, then the CD4 co-receptor would have bound next to it, and instead I'd have CD4 there in this complex. Okay, right. So an interaction like this forms, and what's now going to occur is that the double positive thymocyte receives a signal. So this activates an intracellular signaling cascade which delivers a survival signal. So this is delivering a survival signal to the double positive thymocyte. And this survival signal tells the double positive thymocyte that it can survive. It gives it the signal not to die, not to commit apoptosis. So the tendency is for a double positive thymocyte to now commit suicide in the cortex unless it's told otherwise. And this one here has received this survival signal, so it doesn't commit suicide. Other double positive thymocytes that have T cell receptors, they've designed T cell receptors that unfortunately don't form complexes like this, i.e. they can't bind to either MHC class 1 or MHC class 2 at all. They will die, and this process is known as death by neglect. So if they don't receive the survival signal, they die. So overall, that is the process of positive selection, and I hope you understand what that is actually achieving. That is getting rid of these double positive thymocytes, which unfortunately have designed T cell receptors that are not capable of binding to either MHC class 1 or MHC class 2. Okay? Now, the other part of this signaling process here is to now make this thymocyte here stop expressing both CD4 and CD8. Okay, we want to turn it now single positive. You see, once this interaction has formed here, once we've worked out whether our T cell receptor binds to MHC class 1 or MHC class 2, we can then get rid of the other co-receptor that we no longer need. You see, this T cell receptor bounds to MHC class 1, and therefore the only co-receptor that it requires is CD8. It does not require CD4. It has no use for CD4. So we might as well get rid of CD4 molecules on its surface now. So what's now also going to happen as part of this intracellular signal, that's part of the survival signal, is the T cell will work out which of the CD4 and CD8 molecules it needs. In this case, it needs CD8, and it will stop expressing the other one, and it will then gradually destroy the molecules that it's already produced of the other one. So what will actually happen following this survival signal is it will become what we call now a single positive thymocyte, so an SP thymocyte. So these thymocytes in the cortex, they're going to become single positive thymocytes, i.e. they're only going to express the relevant co-receptor that they need now. So if their T cell receptor binds to MHC class 1 peptide complexes, then they'll keep the CD8 and they'll become CD8 positive thymocytes. And if their T cell receptor meanwhile binds to MHC class 2 complexes with a peptide fragment, then they'll keep the CD4 co-receptor, and they'll now be called CD4 positive thymocytes. Okay, so that's why it's rare to find a cell that has both CD4 and CD8, because generally you only need the one that's actually relevant for your T cell receptor. Okay, the other thing to say is that at this stage in the cortex, there is a process of negative selection as well. So the corticoepithelial cells, not only do they participate in positive selection, but they also do a bit of negative selection. Now, negative selection is going to majorly occur in the medulla, and we're about to come on to that. But in the cortex, there is a degree of negative selection as well. And here is the basic thing that you need to understand. These corticoepithelial cells, they have put these MHC class 1 and MHC class 2 complexes on their surface, and the peptide fragments there are self-peptide fragments. They're peptide fragments from these corticoepithelial cells. So we want the T cell receptors of the thymocytes to bind weakly to one of these MHC complexes. Okay, because if it doesn't, then the T cell receptor is just useless. It's not recognizing MHC complexes. But we do not want it to bind too strongly, because if it binds too strongly, that's not good. That means that it's likely going to be activated by that MHC complex with that peptide fragment in, and that's not good at all, because we don't want to launch an adaptive immune response against that peptide fragment that the corticoepithelial cell is presenting. That would trigger an autoimmune response. So what we now want to do is kill the thymocytes here that actually have T cell receptors which bind too strongly to MHC complexes with self-peptide fragments. So you have to jump through a hoop basically here. You have to jump not too low and not too high. In fact, that's quite a good analogy. There's a hoop basically that you have to jump through, and if you jump too low, then you don't pass positive selection. If you don't bind at all, really, then that's analogous to jumping too low, and then you'd be destroyed by positive selection. And if you jump too high, you bind too strongly, then what's going to happen is the corticoepithelial cell is going to deliver a very different signal to the thymocyte here, and it's going to get the thymocyte to commit suicide, to commit apoptosis. So that's the portion of negative selection is actually going to occur in the cortex of the thymus. Okay, so both positive and negative selection occur in the cortex of the thymus. What's now going to occur is we're also going to have another negative selection, another round of negative selection, which is going to occur in the medulla. So if we've now passed this, so let's say we now have a single positive thymocyte now that has passed both the positive and negative selection processes that have gone on in the cortex of the thymus. So it did bind to an MHC complex of either the type 1 or the type 2, and it didn't bind too strongly to any of them. It survived. What now happens? Well, what now happens is the single positive thymocyte moves from the cortex of the thymus gland into the medulla of the thymus gland. So again, reminding you of the different layers here. Underneath the cortex we have the corticomedullary junction, and then underneath the corticomedullary junction, then in the center we have the medulla of the thymus gland. So what's now going to occur is our single positive thymocyte is going to go down into the medulla of the thymus gland. And in the medulla there are going to be very analogous cells to these corticoepithelial cells. But these are now called medullary epithelial cells, so it all makes perfect sense. Now there are other cells also in the medulla that are going to perform the same role as the medullary epithelial cells, and these are known as the medullary dendritic cells. So we have in the medulla of the thymus gland two cell types that are now going to perform the same negative selection process for the single positive thymocytes that have survived the first two rounds of positive and negative selection in the cortex and are now coming down into the medulla. So the medullary epithelial cells, and we're also going to have, whoops, I've missed our word, medullary dendritic cells. So medullary dendritic cells. Okay, so the picture of these will be pretty much identical to this. In fact, I wouldn't change the picture at all, so I won't redraw it out. They will again have these long processes to increase their surface area, and I'll stress now that the reason that they have those processes is so that they can interact with as many of the thymocytes as possible, because that's, their surface is where they're actually performing their role. That's why they have these processes, to extend their surface area. Okay, so we have medullary epithelial cells and medullary dendritic cells, which look exactly like this. And again, these cells are going to put on their surface MHC class 1 complexes and MHC class 2 complexes with peptide fragments in. And again, these peptide fragments are going to be self-peptide fragments. And you might say, well, what on earth is the point of this? It's just exactly the same as before. No, there is a difference here, and I'm about to tell you what that difference is. The difference here is that these cell types, these medullary epithelial cells and these medullary dendritic cells, they have expressed in them a very special protein known as the AIRE protein, which is known as the autoimmune receptor protein. So the AI is for autoimmune, and the RE is then for receptor, and it's usually just abbreviated to AIRE. Okay, now what does this protein do? Well, this protein, through mechanisms that are still not particularly understood, results in these cells expressing proteins from all over the body. Okay, so the cortical epithelial cells, they had a very limited protein expression. They expressed a few proteins that were important to themselves. They chopped those proteins up and put fragments on their surface. The difference with these ones are that they're going to express an absolutely huge number of proteins. So proteins that would never be expressed in most cells of the body. So for instance, the classical example people always give is insulin. Insulin is not produced by many cells in the body. It's produced by the beta cells of the islets of Langerhans. Most other cells, they have the insulin gene, of course, but they do not express it. The epigenetics is turned off. They do not actually produce insulin. The cortical epithelial cells will not produce insulin, and therefore they will not put fragments of insulin proteins on their surface. These ones will now express insulin, and they will break it up, put fragments on their surface, and this is now really important. So in the medulla of the thymus gland, you're going to have these cells which are going to be expressing proteins from all over the body. And of course, this is absolutely essential. We need to make sure that our single positive thymocyte, the T cell receptor on its surface, it must not bind too strongly to an MHC complex with a peptide fragment from a self-peptide. And so far we've only checked it for a very small number of peptides, the peptides that the cortical epithelial cells produced. We now need to do it for as many proteins in the body as we possibly can, and that's what the medullary epithelial cells and the medullary dendritic cells are going to do. So these cell types are going to express proteins from all over the body. They're going to cut those proteins up and put fragments of those proteins on their surfaces bound to MHC class 1 and MHC class 2 proteins. These single positive thymocytes that have passed the selection process up in the cortex now come down into the medulla, and if their T cell receptors bind too strongly to whichever MHC complex the T cell receptor likes to bind to, and of course it will now have a preference, with a peptide fragment from one of the self-peptides that is expressed in these cell types, then that cell will receive a signal to die. i.e. it's another process of negative selection. You must not bind too strongly. In order to pass the test, you have to get a negative result. You must not bind too strongly. Hence why it's called negative selection. And this, of course, is the major mechanism by which we make sure that we do not produce T cells that are autoreactive, that are going to respond and be specifically targeted against proteins of your own body. Okay? Right, so that's the second round of selection that is going to occur in the medulla, and that's a round of negative selection. Okay, so now we've done all the checks. These cells that finally pass all of that, and they'll have to remain in the medulla for a long period of time, being checked over and over again against peptide fragment after peptide fragment after peptide fragment before we are satisfied. Once they've been in the medulla for a long time and they've been checked and checked and checked against loads of different self-peptide fragments, what will then finally happen is this single positive thymocyte will be allowed to leave the thymus gland, and to do this it will go back up to the corticomedullary junction. So just bringing this picture down, it'll go back up to the corticomedullary junction, which remember is the site of these high endothelial venules. So I'll just draw one of those on there. So here is a high endothelial venule. So now you'll go back up from the medulla into the corticomedullary junction. You'll enter the bloodstream again via these high endothelial venules in the corticomedullary junction, and now you are a mature T cell. Now let me introduce you to a bit of nomenclature before we then have a break. So T cells that have T cell receptors which can bind to MHC class 1 complexes with a peptide fragment on, and which are therefore CD8 positive. Remember, CD8 is the co-receptor for MHC class 1. So CD8 positive T cells, as we call them. These have another name which I want to introduce you to now. They are also known as cytotoxic T cells, or just as T subscript C cells. And these are the T cells that are going to be involved in the cell-mediated adaptive immune response. So these are the ones that are going to be responding to intracellular pathogens. So it all hopefully is starting to make sense. Remember, intracellular pathogens, they get their peptide fragments loaded on MHC class 1 protein complexes. MHC class 1 protein complexes are going to be activating these CD8 positive T cells, and these, I'm now telling you, are also called cytotoxic or T subscript C cells. And these are going to be involved in that cell-mediated response, killing cells that are infected with the intracellular pathogen. Meanwhile, the CD4 positive T cells, these are the ones with T cell receptors which can bind to MHC class 2 protein complexes with a peptide fragment in them. These are also known as helper T cells, and these are going to be involved in the humoral adaptive immune response, the adaptive immune response to extracellular pathogens, which results in the production of antibodies and the activation of B cells, as we'll understand later on. And helper T cells are also known as T subscript H cells. Okay, and you'd read this as helper T cells. Okay, so we have two major populations of T cells then. The CD8 positive T cells, or cytotoxic T cells, and the CD4 positive T cells, or the helper T cells. And you would refer to the things that we are producing now as mature CD8 or positive or CD4 positive T cells that are leaving the thymus gland. If you want to give them their absolutely full name, you'd give them the name, they are mature naive T cells. They're naive because they have not been activated so far, they've just been produced. Okay, so they are mature naive T cells. We will have a break there, and in the next video what we will do is we will explore what's going to happen to these mature naive T cells after they've actually been produced. Don't worry, I haven't forgotten about explaining what happens in the adult after the thymus has involuted. We will come back to that later on. Okay, so we'll have a break here, and in the next video we will discuss where are these mature naive T cells of the CD4 positive and CD8 positive type going to actually go, and we'll see that they're going to circulate between the circulatory system and the lymphatic system.